Thank you.
Okay. Good morning to everyone. It's a great pleasure for me to start this session this morning. Basically, before starting everything, I have really to thank again Patrick and Biet for their really wonderful organization and wonderful meeting, conference. And actually, people that came from my lab and other labs that I'm associated with, all are very happy that they are here because they claim really this meeting opened for them new world. And that's actually one of the main purpose of a meeting like that. It's probably one of the most interdisciplinary meetings that you can find on the globe, focused on one topic of nanomedicine, nanotechnology, but basically from many, many perspectives, and I think this is unique. Well, saying this, I actually thought for a long time that nanomedicine, which is based on nanotechnology, which is based on a lot of physicochemical methods, it's about time to talk a little bit about these physicochemical methods. And can you start the slide, please? And that's why I discussed with Biet to try to organize this meeting. And I think the way we are going to do it, we are going to have, I'll give a very, very short introduction in the beginning, and then we'll have a few topics. Actually, it seems that some people did not show up, and that's, well, it has a bad side that not all the topics are covered, but it also has a good side that we can talk a little bit more in depth about some of the issues. One of the people that did not show up is Peter Jimson. He is supposed to cover the calorimetric aspect, the thermodynamics. So I will take over, and he gave me his lecture, which I assembled with some recent observation we made using these approaches. And I think I'll keep it as a kind of a secret at the end and show you some really, what I think really very puzzling and interesting data, very recent that we never discussed in anywhere yet, using DSC, using the calorimetric approach to characterize nano-drugs. So actually, we are going to talk about key characterization methods. I really brought an excellent team of speakers today, and we'll cover people that are here. We'll cover Scott McNeil from the NCL will cover general characterization, and I want him to focus a little bit about size measurement, which are very, very critical to the nano, as we all know from lectures that were discussed here. Tony Watt didn't come, unfortunately, so I don't know if then we'll have to miss the NMR, sorry. Uri Raviv from Jerusalem University will talk about X-ray diffraction, a very, very powerful technique. And then Oren Regev from Ben-Gurion University will talk about cryothem, quantitative cryothem. Again, I think it's a very exciting. I have the opportunity to work with these guys, so I can tell you that it is really exciting story. About Scott, I told you, and then I'll be the last one talking about calorimetric measurement, and then we'll have some examples from different people that I asked to bring them and show up. And then we'll have enough time for questions and answer or debate or whatever you want. I don't think there is much to debate here, but there is a lot to talk about. So why is this thing so important? If you look at what we're dealing with nano, and this is not a complete a kind of a system, but you see many, many systems. They are all share one thing. They are all in the nano range. And by this, they also share the ability to take advantage of the EPR effect if they are good enough to benefit from it, namely if they can circulate long and if they are at the right properties, then they can benefit from the EPR effect. We still face with another problem, which I'll talk in my second lecture later in the afternoon, about how drugs are released once they get into the site of inflammation or site of the tumor. But as you can see, these systems are very different one from the other, and they have very different physical-chemical properties. We should take advantage of these differences and understand them well, and the only way to understand this difference is apply some of the method and maybe some other method that we'll discuss in our presentation today. Actually, if we talk about nano-drug, as I said, there are many nano-drugs, and we have to be really being capable of analyze what we have. This becomes more obvious when I show you what FDA required of us when we're dealing with nano-drugs. Actually, the list is quite amazing of how many things. And, you know, after really having quite a large experience in this field, I can tell you that the FDA is absolutely right. We'll go into it in a minute. So if we're dealing with the nano-drugs, they are by definition, they are all super assemblies. They are very complicated structures. And we really have to know how to characterize them. And that's required a lot of understanding in the meetings, in the methods we are going to discuss. Also, a very important aspect is to understand the crosstalk between these physical-chemical properties, nanotechnology, and the biological principle, like what happens when you have inflammation, what kind of microenvironment you have. All these are very critical to interact with the physical-chemical properties. So just the physical-chemical property by themselves are not sufficient, but if you try to understand how they interact with the biological milieu relevant to the disease you are going to deal with, then you can benefit from it a lot. This just to give you a feel what the FDA required. This is actually come from the draft guidelines. One thing I wonder why FDA has only drafts all the time, and it takes a long time. By the time they come with the real guideline, the product of is no interest anymore. So they have guidelines about draft guideline, which was updated. This is about generic Doxil, but we can learn a lot about this, what the FDA requiring from us regarding the characterization of system. And if you read carefully, there is the biochemistry, very careful biochemistry in terms of what are the components that compose the things, your carrier, your system, how much encapsulated drug you have, all these things. And then if you look carefully at the second line, they said lipid bilayer phase transition. You can see it here. And that's a very, very important, as I'll show you later, and there are quite a lot of surprises there because people don't use it very often. And I think this use is a must, and I'll show you later why. Size distribution, really, I don't think I have to say much about it, except that it looks simple, but it's very, very complicated to get the right size. I hope Scott will talk about it a little bit. We have a lot of methods. Everyone come up not with the same results, and I always wonder what is the right size of the particles we are dealing with. So we leave it for the discussion later. Then in the case of a system that have internal environment, we have to deal with measuring the internal environments. Let's say, what is the internal pH? In the case of ammonium sulfate gradient, what is the gradient? And what is the structure, how it affected the bilayer. And here come methods, very powerful methods like the X-ray diffraction that we are going to talk about. Small angle and wide angle, both are very important and give us information you cannot get by any other method. You have to remember. I think then you have to deal with the surface, to measure the surface, and one of the requirements that we are going to talk about today in the case of pegylated system is to measure the size or the length of the PEG layer and its distribution. Again, here X-ray seems to be one of the most, first of all, the direct method and a very powerful method. And one of the things that you have to ask really in the case of generic or similar or biosimilar is really to do the comparison with all this method. And I think in this case, the requirement, all the requirements of FDA are justified. I think that this, as I show you, and other people will show you, these physical-chemical properties are really critical to the performance of the particles, and therefore I think they represent really the right requirement that we have to come with in order to really know what we are dealing with. And then we have to deal later with the in vivo, in vitro correlation, how all these physical-chemical properties correlate with what's happening in vivo. And that's a very important aspect to deal with. So I think I already said what is written here, and these are the methods, and I'm looking forward for a very exciting session. And I want here now to get our first speaker, which is present here, and this is Uri Raviv, which will talk about X-ray diffraction, the different aspects of X-ray diffraction. Uri, please.